can you believe it? We're actually going to finish genetics. This mini lesson, that's it. No, it's kind of a bummer because genetics is pretty stinking cool. But today, well, tonight, or whenever you're watching it, is actually one of my favorite things to talk about because it's kind of, well, it's kind of the big picture genetics, but it's kind of why people really get hooked on genetics and study it so intensely. So I do want to kind of go over the big picture. And I found this picture, of course, of a big picture. That's a pretty big picture. Anyways, why study genetics? I just want you guys to think about some of these questions that I bring up. Why study genetics? Are you interested in it? Do you like knowing about your kind of history and what gets passed down and how things kind of play out when it comes to, offspring and such? Why does it interest you? Think about these things. How does the study of genetics affect you and the people you know? These are kind of what I think of as the big picture of genetics, why we even talk about it in biology. But these things are really kind of cool to think about because they involve you personally. In biology, the study of life really does involve you because we are living, but this is really cool because you get to see why you are you, okay? So anyways, just a little kind of thing to think about. So what we're going to talk about is a bunch of different things, and I believe this is the last section of genetics, biotechnology, okay? The first thing that you'll see in this section is biotechnology. This is the newest ways of changing species collectively, okay? So basically, how we change species collectively, biotechnology. And when you think about that, you're like, what? Like, like we're doing this? Like we're going to have a life-size rooster? Well, who knows? So anyways, I want you guys to think about that. That we have the ability to do these things now, which is kind of scary, kind of crazy. But let's go a little bit further. So I've got one thing to show you. Well, I've got many, of course, you guys know, many videos to show you. Kind of a humorous one. So watch this and see if this is what we're in for.
Old MacDonald had a farm, E-I-E-I-O. And on this farm he had some chicks, E-I-E-I-O. With a chick here and a chick there. Here a chick, there a chick, everywhere a chick. Old MacDonald had a farm, E-I-E-I-O. Old MacDonald had a farm, E-I-E-I-O. And on this farm he had a cow, E-I-E-I-O. With a moo here and a moo there. Here a moo, there a moo, everywhere a moo, moo. Chick here and a chick there. Here a chick, there a chick, everywhere a chick. Old MacDonald had a farm, E-I-E-I-O. Old MacDonald had a farm, E-I-E-I-O. And on this farm he had some ducks, E-I-E-I-O. With a quack quack here and a quack quack there. Here a quack, there a quack, everywhere a quack quack. Moo moo here and a moo moo there. Here a moo, there a moo, everywhere a moo moo. Chick here and a chick there. Here a chick, there a chick, everywhere a chick. Old MacDonald had a farm, E-I-E-I-O. Old MacDonald had a farm, E-I-E-I-O. And on this farm he oink here pigs, E-I-E-I-O. With an oink oink here and an oink oink there. Here an oink, there an oink, everywhere an oink oink. Quack quack here and a quack quack there. Here a quack, there a quack, everywhere a quack quack. Moo moo here and a moo moo there. Here a moo, there a moo, everywhere a moo moo. Chick here and a chick there. Here a chick, there a chick, everywhere a chick. Old MacDonald had a farm,
Oh boy. All right. So kind of a joke, kind of not. Here's the thing. When we talk about what we're going to talk about in this lesson, you have to understand there could be some serious consequences for some of the things that we're doing. So I want you guys to start thinking for yourself. I'm going to present to you some information. See what you think about that, okay? Start to develop your own opinions. But here, let's talk about some of the things that you're going to read about in your lesson, genetic engineering. So genetic engineering, also known as recombinant DNA technology, kind of a long word, but those are kind of go hand in hand. They're the same things. Means altering the genes in some living organism to produce a GMO, which is a genetically modified organism that has a completely new genotype, okay? So it's kind of interesting that you take one organism with a genotype and you can actually alter those genes to have a new genotype. Kind of crazy. And sometimes even me as a science teacher, I think about that. I'm like, wow, we're really capable of doing that, right? That's crazy. So genetic engineering, not that they're really, I mean, some of the pictures I show you are going to be a little exaggerated, so it's not like they've come up with this crazy orange that has an apple skin, but you never know. It might happen soon. So that is what, first of all, know that GMO, genetically modified organism, you're going to see that every now and then, and you're going to hear about that. But know that genetic engineering is taking, altering those genes in a living organism to produce a new genotype, okay? Not a new organism necessarily, but a new genotype within the organism, okay? Various kinds of genetic modification are possible. There are such things as inserting foreign genes from one species into another, forming a transgenic organism, which we're going to talk about in a little bit, altering an existing gene so that its product is changed, or changing gene expression so that it is translated more often or not at all. So all of those things are different types of genetic modification. Some are really good, some are really beneficial, some could really hurt us in some ways, okay? We're going to talk about some of those, okay? Recombinant DNA, and I say recombinant, what? You're like, huh? What is that? Okay, it's a form of DNA that contains segments that would not normally exist together. Okay, keep that in mind. So recombinant DNA would not be possible without the discovery of restriction enzymes. So you need a couple of things for this recombinant DNA. Remember, recombinant DNA, a form of DNA that contains segments that would not normally exist together. Okay, keep that in mind, not normally exist together. So you need restriction enzymes. These enzymes are going to create DNA fragments, so they're going to cut it at certain base sequences. And these restriction enzymes attach the DNA molecules at specific sites, and they cut both strands of the DNA at that place. Okay? All right. Try and picture this. Try and imagine this. Okay? That's what's happening with recombinant DNA. You need those restriction enzymes. The basic steps in genetic engineering. So remember, you need that recombinant DNA, but I'm going to go kind of through the steps real quick. So isolate the gene and treat it with a restriction enzyme. Okay? So you have a gene that maybe you want to alter. Maybe there's some gene, and we'll use some examples in a bit, but say, for instance, like crops, that some kind of gene that makes them really appealing to certain pests or bugs, and you want to take that gene and you want to treat it somehow. You want to fix it. Okay, so you're going to isolate that gene and treat it with a restriction enzyme so the DNA is cut. Okay, insert a host vector. Okay, insert a host using a vector. A vector is a molecule of DNA which is used to carry kind of a foreign gene into a host cell. So I always think of like a vector when you're kind of being taken over by a virus or something, okay? You're getting one of those cold viruses, and what they do is they insert those genes into your cells, and all of a sudden you're sick, okay? So I kind of think of that as like a vector. A vector is a molecule of DNA, though, which is used to carry a foreign gene into a host cell, okay? So it kind of inserts that foreign gene into the host cell. So it produces many copies of the host as possible. That's what's happening in that genetic engineering, is once you have that vector that has inserted that foreign gene, it's going to reproduce and make as many copies as possible. And then you're going to separate and purify the product of the gene. Okay? Easy enough, right? So here's an easier way to show it. It's the same that you'll see in your lesson. I really liked this diagram because I thought it was fairly cut and dry. So step 1, cutting the DNA. DNA is cut from the organism containing the gene of interest in the vector, which is the agent that is used to carry the gene of interest. All right? Step 2, making recombinant DNA. The DNA fragments from the organism containing the gene of interest are combined with the DNA fragments from the vector. The DNA fragments bond together with the help of an enzyme known as DNA ligase. Okay, so that's where we're making that recombinant DNA. Step 3, cloning. In a process known as cloning, many copies of the gene of interest are made each time the host reproduces. Okay, so we talked about that in my last slide, but that's referred to as cloning. And then step 4, screening. Cells that have received a particular gene of interest are separated. The cells then can transcribe and translate the gene of interest to make the protein coded for in that particular gene. Okay, so that's genetic engineering in a nutshell. You have those 4 steps. So look at this again in your lesson and go through that and know those steps, because that's kind of the basis of genetic engineering. So if you know that, go ahead, try it yourself. Just kidding, not that easy. Okay, so how about some agricultural applications? I kind of talked about a little bit. Pest resistance is one big one. So insecticidal proteins have been transferred into crop plants to make them pest resistant. So we all know that, farmers don't want to lose their crops because of pests, so they're spraying everything. That's one thing. But then all of a sudden you're getting people that are like, I'm not buying that stuff. Not with that pesticide. That's harmful to my body. So guess what? Now they're able to change that gene or basically transfer that gene into the crop plants to make them pest resistant. Sounds like a good thing, could be a good thing, but remember, we are also disturbing kind of the circle of life. So anyways, I'm going to lead you to have your own opinions about all that, and I have mine as well. Here's a good thing, golden rice that's been genetically modified to produce beta carotene, which is provitamin A. So converted into the body to vitamin A. This is something that was absolutely needed at a time where vitamin A was depleted, okay? And people needed that. So it's interesting that golden rice was basically created and has kind of served a major purpose to getting people the right amount of vitamins and minerals, okay? So, in this particular instance, vitamin A. So that's a great thing. There's a lot of good things to, the genetic engineering. Adoption of genetically modified, which we call GM, crops has been resisted in some areas because of questions about. So here are some of the things that you might think about. Crop safety for human consumption. Is it safe? We don't know. We're the guinea pigs. Okay, movement of genes into wild relatives. Okay, interesting statement. Loss of biodiversity. So basically we're kind of making everything the same. There's no diversity. And that's, I want you guys will probably be like, yeah, but whatever. I go to the store and I see apples and oranges and bananas and all, that's biodiversity. But we're talking about within one species or one organism, okay? We lose that biodiversity. So think about some of those things, because those are some of the questions that people bring up, okay? How much do you know about genetically modified organisms, okay? Let's look at a quick video and see what other people think or know.
So do you know what genetically modified foods are?
Like tomatoes and things they do like to make them last longer when they ship them and stuff.
Right.
Bettered or improved by collecting the best of and keep going. But
I've heard of them. Yeah, I don't know how big of a fan I am, but...
Right.
Yeah, I don't really know if it's good or bad.
You know, we've been eating these foods for the last 10 years.
No, I haven't known that.
Well,
Where do they normally sell those kind of foods?
I'd kind of taken back by it. I thought it was just grown in soil, and I didn't know they were modifying my food.
I know.
It's kind of scary.
Well, I know it's been a while. I didn't realize quite that long.
well, the simplest way to put it is that scientists had an idea to make crops more resilient to external factors. So let's say you grow tomatoes, and at night it gets so cold that by morning half your tomatoes are frozen, and now you can't sell them anymore. This is what scientists were able to change through genetic modification. They figured out a way to take DNA from one organism and put it inside a completely different organism to create a desired and specific outcome. For example, let's go back to the tomato scenario. Scientists recognized that Arctic flounder, yes, that's the fish, could withstand extremely cold temperatures. They thought, what if we took the DNA trait that allows the flounder to withstand the cold and put it inside the tomato DNA so that the tomato could withstand the same temperatures? Now you've just narrowed your chances of your tomatoes becoming frozen. This idea that you can now get 100% crop yield was the original guiding principle for genetic modification. Our friends at Lilly Films have a documentary called The Future of Food by Deborah Coons Garcia that explains it best.
Genetic engineering is really a radical revolution in food production. It's really a cell invasion technology. You know, people have heard they're taking a flounder gene and putting it in tomato so the tomato can last in cold temperatures. But people ask, how does that flounder gene get in that tomato? How does it get in there? And what really happens is, the only way you can do it is to invade the cell of the tomato and deposit the flounder gene. Well, what's good at invading cells? Bacteria and viruses.
After 12 years of searching, Monsanto found a soil bacteria that is naturally immune to Roundup herbicide. Their goal was to genetically engineer DNA from these bacteria into various plants. They cut out a sequence of DNA that is resistant to Roundup. But if this DNA sequence alone is inserted into a corn plant, it will have no effect. So the next step involves E. coli bacteria. Gaps are created in the E. coli DNA, and when the two test tubes are mixed together, some of the E. coli DNA recombines with the Roundup-resistant bacteria. Then the technicians smuggle the engineered DNA into the cells of the corn plant they want to modify. Cells will naturally reject foreign DNA, so they developed a method using soil bacteria that causes tumors in plants. They use this bacteria to ferry the engineered DNA into the plant's nucleus. There are also two other methods used to get the engineered DNA through the cell wall. One uses a stream of electricity to create tiny holes in the plant cells so they become vulnerable to infiltration by foreign DNA. Another is the gene gun, which blasts particles of gold coated with engineered DNA into the plant cells. Each of these three methods needs a promoter gene that turns on the desired characteristics. The promoter gene is often extracted from the cauliflower mosaic virus.
This capacity of bacteria and viruses to invade mammals in different ways is what really has a lot of people edgy about biotechnology, because that's really what the tools are all about. In order to move genetic material from one organism to another that don't normally cross, you've got to sort of behave like bacteria and viruses and invade into the cells and become established, just like the virus must become established.
American Heritage Medical Dictionary defines genetically modified organisms as an organism whose genetic characteristics have been altered by the insertion of a modified gene or a gene from another organism using the techniques of genetic engineering. The hope was that this biotechnology would end world hunger by creating higher crop yields. Ironically, we have found studies that indicate just the opposite. Reports have shown that genetically modified crops actually produce fewer yields. In fact, in some countries where starvation rates are very high, there have been many cases of citizens burning genetically modified crops in protest. Like the Future of Food said, Monsanto, the largest biotech company in the world, was able to make seeds that resisted their own Roundup herbicide. This allows farmers to spray as much herbicide as they want without worrying about their crops dying. But it doesn't stop there. Scientists can now make seeds that produce their own pesticide. This is like you and me walking around with mosquito repellent coming out of our pores. How do they do this? Scientists found a bacteria in the soil that is lethal to many species of insects called Bacillus thuringiensis, or BT for short, and they genetically engineered it into the crops. So what could the effects be when the bacteria that only used to live in our soil is now being distributed into the air through our crops? In the Philippines, the people living next to the genetically engineered cornfield developed recurring skin, respiratory, and intestinal problems. Blood tests were done on 39 of the Filipinos, and it was revealed that their immune system was affected by the BT toxin that the corn had released. And there's still more. Instead of just making drugs in labs, scientists have been growing crops that produce their pharmaceutical drugs, like research chemicals, blood proteins, antibodies, and vaccines, just to name a few. According to the Society of Diabetic Rights, thousands of diabetics have experienced bad reactions, and many have died as a result of this genetically engineered insulin called synthetic or human insulin. The Union of Concerned Scientists have been urging the United States Department of Agriculture to put a ban on pharma crops due to the potential that they could contaminate all of our food supply.
Okay, so that was kind of a long video, but I really wanted you guys to see that. Now that is one side. You're going to see other sides that will only promote GMOs, okay? So I want you guys to take that in. I'm kind of one of those naturalists that I would prefer everything to be just plain, normal, nothing genetically modified. But some of you guys may have different opinions, and I want you guys to know that this is a class where everyone's opinion is respected. So that's just one side, but please go and find some other things if you like. That if you're totally for that, there are plenty of videos out there that are like, yeah, let's keep this up because blah, blah, blah. All right? So anyways, let's move on here. USDA organic. The USDA now certifies all food that has an organic label to be tested, okay? So you can take an organic, that's something that's marked organic, have it tested. Yes, there's nothing genetically modified about it, okay? So here's one last video that I'm going to show you guys, and then we'll kind of breeze through the rest of the stuff. One of my favorite movies, and some of you guys have seen it, well, it's not my favorite, but it's a good documentary, and I think everyone should see it. There's many of kind of different documentaries out there about something like this, but this is called Food Inc. So if you haven't seen it, definitely, I don't think it's biased. I think it's definitely telling the truth, but you need to know what's going on with some of our food products.
The way we eat has changed more in the last 50 years than in the previous 10,000. The modern supermarket has on average 47,000 products.
The industry doesn't want you to know the truth about what you're eating, because if you knew, you might not want to eat it. We've never had food companies this powerful in our history.
Everything we've done in modern agriculture is to grow it faster, fatter, bigger, cheaper.
If you can grow a chicken in 49 days, why would you want one you got to grow in 3 months?
When you go through the supermarket, there is an illusion of diversity. So much of our industrial food turns out to be rearrangements of corn.
Sometimes you look at a vegetable and say, okay, well, we can get 2 hamburgers for the same price.
They have managed to make it against the law to criticize their products.
There is an effort to make it illegal to publish a photo of any industrial food operation.
I find it incredible that the FDA wants to allow the sale of meat from cloned animals without any labeling.
Peanut butter contaminated with salmonella.
E. coli has been found in spinach, apple juice.
Smells like money to me.
The average consumer does not feel very powerful. It's the exact opposite. When we run an item past the supermarket scanner, we're voting for local or not, organic or not.
Look at the tobacco industry. The battle against tobacco is a perfect model of how an industry's irresponsible behavior can be changed.
Imagine what it would be if, as a national policy, the idea would be to have such nutritionally dense food that people actually felt better, had more energy, and weren't sick as much. You know, now see, that's a noble goal.
People have got to start demanding good, wholesome food of us, and we'll deliver. I promise you.
Okay, that's a really good documentary, and I really encourage you guys to watch Food Inc. If you have Netflix, it's a watch instantly. But I think so much of this genetically modified, all the stuff that's going around, you just have to be aware. You have to be educated. You have to know what you're eating. You have to demand quality because, again, most of us are in this fast-paced world and we just want cheap and inexpensive and fast. And so therefore, the people that are making our food are going to make it cheap and fast and unhealthy because that's what makes it cheap and fast. So watch that, please. I'd love to hear your feedback on that because it's a great movie. But moving on. There's such thing as a recombinant DNA vaccine. Huh, interesting, right? Strategy for a subunit vaccine for like herpes simplex is one example. So basically you have the DNA that's extracted from this herpes simplex. The gene is isolated. The genes, I'm trying to read this, sorry, it's very small. Gene specifying herpes simplex surface protein. And basically it is then inserted in here. It says harmless engineered virus, the vaccine with the surface like herpes simplex injected into the human body. Okay, and then antibodies are made to basically protect you from that. So that's kind of what a vaccine might look like. And there's a lot of talk about vaccines these days about whether or not they're good for you, but that's a whole nother topic. We won't go into that right now, but basically they use kind of the same recombinant DNA type thing when they have certain vaccines, okay? Kind of interesting. And then you have something called gene therapy. Gene therapy. So one example, the treatment of SCID, which is severe combined immunodeficiency. It affects the maturation of immune cells that develop in bone marrow. And then these type of people that have SCID suffer lacking the enzyme ADA, which is adenosine deaminase. Okay, so basically you can use gene therapy. You remove bone marrow, which are stem cells. Okay, if you guys didn't know what stem cells are cells that have not specified themselves. They could turn into muscle cells, they could turn into bone cells, they could turn into nerve cells. And you find these stem cells within the bone marrow. Okay, so you can take these stem cells basically out of the bone marrow before they've become anything else. So they're kind of this, I don't know what I'm going to be yet kind of cell. All right, and you have this defective gene here that's causing the SCID. And you use these retroviruses to bring the normal gene into the bone marrow of these stem cells. So basically you take this retrovirus with the good stuff, kind of, you have this defective gene where you have now the good stuff. You want to replace it kind of. So you have that viral recombinant DNA in that normal gene, and then the recombinant DNA carries the normal gene into the genome. Okay, and then you have all these different things that happen, reverse transcription, blah, 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 but ultimately you return that back in to the bone marrow, and in hopes that you have kind of reversed that or stopped that gene from being reproduced, okay, this defective gene. So that's gene therapy. Kind of cool. I mean, there's obviously some great things to all of these genetic studies, okay, what they're doing with this to help people either kind of make it through some of their diseases or completely reverse them, okay? So, kind of interesting. So, analyzing DNA segments via gel electrophoresis, okay, electrophoresis. DNA can be subjected to DNA fingerprinting. And you guys will read more about this in your lesson, okay? There's a lot to this. But what we were talking about is basically you can treat a DNA segment with restriction enzymes, okay? Remember those restriction enzymes we talked about when we were discussing recombinant DNA? So what happens is a unique collection of different fragments is produced. The gel electrophoresis separates those fragments according to their charge or size, okay? And I'll show you a picture in a second. And produces a distinctive banding pattern. So usually we use these to measure numbers of repeat or short sequences, okay? And they're used in paternity suits, rape cases, corpse IDs, etc. So you can use this gel electrophoresis to look at a DNA segment and kind of see who this is and pinpoint, who's the father and who did this in this rape case. And, if you can't identify a corpse by their face, then you can do it by using the DNA and doing this electrophoresis. Okay, and this is what kind of a picture looks like. So, you'll see that a lot when you hear about CSI, that they'll have to do something like this to get the DNA from a suspect or something like that. So it's a technique used to separate DNA fragments by size. The gel, which is typically this agarose or polyacrylamide, which I'm not sure if I'm saying that correctly, but these are subjected to an electrical field. The DNA then, which is negatively charged, migrates towards those positive poles, and then the larger the DNA fragment, the slower it will move through that kind of matrix of gel. DNA is visualized using fluorescent dyes. So it's kind of interesting, this whole process. if you take forensic science, you'll learn a little bit more about that. So it's kind of cool, very cool kind of a process. But that's the basics to it. You can also do DNA fingerprinting and paternity. So male 1 is the father. His DNA fingerprints look like that of the child. So here's the DNA fingerprinting of the mother. Here's the child. Who's the dad? Is it male 1 or male 2? So you look at this and you think, what looks most similar to this child? Okay, and you see this pattern here. And you're like, all right, okay, how about male 2? Well, no. Okay, sorry, male 1, you're it. So basically you can look at that and find out who the father is. All right, Human Genome Project, we're moving on. So the Human Genome Project is kind of interesting, identifying and locating approximately 100,000 human genes on all chromosomes, determining the sequence of the 3 billion base pairs in the human genome. This is this project that has been kind of ongoing to help us understand a lot more about our genomes and all the genes that exist within our chromosomes. It's pretty overwhelming when you think about it. So knowing where a gene is located as well as the nature and action of its product will prompt development of new treatments for disorders or particular disorders, okay? So that's kind of what the Human Genome Project is. It's going on currently. They're finding out so much information and they're adding to it daily. So genome, which I've talked about that term before, and I'm not sure how often I've actually defined it, but a genome is all the genetic information of an individual or even a species, okay? So you can have a genome specifically for yourself, but you can also have kind of a basic genome for a species. So the goals of the Human Genome Project completed in 2003, although like I said, they're still finding things daily to kind of add to that, but determine the base pair sequence in all human DNA. I mean, that's quite a task, okay? It was a 13-year project, so it took 13 years to find the base pair sequences in all human DNA. They first had a working draft. They constructed a map showing the sequence of genes on specific chromosomes, and then other species kind of are in their final stages as well. I'm not sure as we speak. I'm not sure if they've completed some of those other tasks, but it's pretty cool to think about it. I'd love to be a part of that. Can you imagine if you're like, yeah, I helped out with the Human Genome Project? You'd be like, oh my goodness gracious, how he's so smart. So this is really cool. This is just a partial map for the human X chromosome. So this is part of the human genome, is that they took each chromosome and basically were able to figure out every single gene and where on the chromosome it's located. Okay, so if you found that there was some kind of a mutation or genetic deletion, whatever kind of occurred in a certain area, you could see what could have happened. So like if something right here, this is pointing to Duchenne muscular dystrophy. What else can I read? Let's see. Down here, Fragile X syndrome. So again, there's a lot of things, obviously tons of different things located on each and every gene. This is just the human X chromosome. All right, and cloning. We're not going to talk a whole lot about that, but think about that. This has been around for quite some time, where you had Dolly the clone, the cloned sheep. But obviously we haven't gotten to the point where we've talking about that with, or we've taken it to the level of cloning humans, at least not that we know of. But kind of an interesting concept to think, of cloning, having an exact replica of a species. Would you agree? Do you disagree? Okay, so that's part of DNA, I can't talk right now, manipulation. Cloning is the production of an identical copy of DNA through some asexual means. So there's no sexual reproduction here. Cloning is asexual, but you're producing an identical copies of DNA in order to make an exact organism, okay? An underground stem or root sends up new shoots that are clones of the parent plant. That's one way of cloning. That's kind of a normal way of cloning. Members of a bacterial colony on a petri dish are clones because they all came from basically division of the same cell. So clones exist very naturally, okay? Human identical twins are actually clones. The original single embryo separated into two individuals. So if you guys didn't know that human identical twins, basically you have one egg and it's been fertilized, so you have a zygote, Okay, so you have that fertilized egg, and all of a sudden it splits in two. That's a clone. So if you're a twin, identical twin, not fraternal, because a fraternal twin is two separate eggs that have been fertilized at the same time. Okay, but identical twins are clones. Kind of interesting, but not like, let's make a clone of you right now. So these are naturally forming clones. So cloning isn't a crazy thing that only happens when you have Dolly the sheep. So I want you guys to know that cloning happens. Gene cloning is the production of many identical copies of the same gene. Clone genes have many research purposes, determining base sequences between normal and mutated genes, altering the phenotype, obtaining the protein coded by a specific gene, and etc. There's many things. Humans can be treated with gene therapy, which is an alteration of that phenotype in a beneficial way. So that's kind of another gene cloning. So there are some benefits to that. So cloning a human gene. So you have to have this restriction enzyme, ECO-RI, bacterial enzyme that stops viral reproduction by cleaving viral DNA. Now this is going to be a little intense, so if you don't get this right away, that's okay. Act as a molecular scissors, cut plasmids and foreign human DNA, produce short single-stranded sticky ends where insertions of foreign DNA can be made. Okay, so you have basically this picture that's a pretty good example of taking human DNA. You have bacteria, you take that plasmid, and basically you have that restriction enzyme right here. It cleaves the DNA. Your DNA ligase seals the human gene and the plasmid kind of together. The host cell takes up the recombined plasmid, and then you clone via kind of these bacteria, which is kind of crazy. All right, so there's cloning of a human gene. So choosing the sex of your baby, yet another way to manipulate genes. So it's interesting that now there are ways that you can do this. You can even go so much as to say, I want my child to have blue eyes. I don't know how much people pay for this stuff, but I'm just curious. Think about what your thoughts are on that. That to me is, taking it out of our hands or putting it in our hands when it shouldn't be. But I want you guys to think about that. Sometimes people go the opposite way and say, but what if there is a gene for a disease that's been passed down and I don't want my children to get that? So you have to take a look at both sides, okay? Do we want to play God and just choose hair color and eye color, or do we want to prevent some of our offspring from having certain diseases? Well, again, that's a choice that you're going to have an opinion on, and so do I, and that's okay. Transgenic organisms, these have been genetically engineered, a breeding approach that uses recombinant DNA techniques to create organisms with new characteristics, okay? So, if you hear transgenic organisms, it's kind of interesting that you're going to have, these organisms with very new characteristics that maybe they never had before, okay? Again, another kind of genetically engineered type GMO, okay? These are identified as a class of genetically modified organisms. So the benefits could be genetic engineering can cure diseases, improve upon existing plants and animals. Such improvements can be natural pesticides, maybe that's a good thing, biotoxin, as well as improved yield crops and animals, although some people beg to differ in that. Problems. What does it mean to improve or make something better? Okay? What is that taking? Are we sacrificing things? I want you guys to think about that. Okay? You gotta read some of these, by the way. What happens when genetically engineered plants or animals escape into nature? Okay? Could change a lot of things in what we know as some of our species. Okay? And then are all of the side effects known? Should they be further studied before research continues? So that's a big thing. So we don't know all the side effects of some of these genetically modified organisms. Could they be bad? Could they be harmful? We're not quite sure. So some moral questions to kind of ask yourself, because I just want you guys to think about this. Should scientists try to make plants or animals better? Do they need to be made better? Think about that. You don't have to answer that. You don't have to tell me that, but think about it. What does it mean to make something better? What does that really mean? Is something not good enough now? Does it need to be better? Should genetically engineered food be fed to animals that humans are going to eat? Okay, that's another thing. Should the government require labels on all genetically engineered foods? Do we have the right to splice animal DNA into our own bodies? Can we become real chimera, animal/man? Scary. Is this the future? You want to be this guy? Yeah. Just kidding, that's kind of scary. All right, so you decide. It's up to you to educate yourself, decide how you feel about this topic, and many scientific topics to come, because we're going to talk about lots of different things that maybe you have your own opinions, and that's a good thing. So that's it for genetics. I hope you guys have enjoyed this unit. It's a great thing to kind of dig more into. If you guys are interested, look into that, taking something like forensic science or studying that more in college. All right, on to Unit 5 next time.